Hey everyone, how are you all? This is Dr. Gayatri. Welcome you all to BioSchool. So this is a continuous session where I'm going to discuss CSIR NET 2020 question paper. So today we'll discuss question from 16 to 30 of part B from morning session. So let's get started. Question number 16. Which one of the following approaches or markers would be typically used for discovering polymorphism between two closely related accessions of a crop plant? Okay, so this is asking about which of the following approaches or the markers that is used for discovering polymorphism. All these techniques are used for discovering polymorphism, but it is asking discovering the polymorphism between two closely related accessions of a crop plant. So the two closely related accessions of a crop plant that can be analyzed by using GBS. GBS is genotyping by sequencing. Genotyping. So this is the most appropriate answer for this question. Right? Sequencing. So if we sequence the two plants, then we can easily distinguish the plants. Okay, so the correct answer is option 2. Question number 17. The rate constant for conversion of a substrate into product. Conversion of substrate into product. The rate constant is 10 to power minus 4. 10 to minus 4. While the reverse rate constant is, reverse rate means the conversion from product to substrate. It is 10 to power minus 6. An enzyme enhances the rate of reaction by 100-fold. So enzyme enhances the reaction by 100-fold for both the reaction because it maintains the equilibrium. The equilibrium constant for enzyme catalyzed reaction is, it is asking the equilibrium constant. So the equilibrium constant, that is can calculate by the rate constant for forward reaction divided by the rate constant for backward reaction. So what is the rate constant for forward reaction? Forward reaction is 10 to power minus 4, and for backward reaction it is 10 to power minus 6. Okay, so subtract this is the forward reaction and this one is the backward reaction. So by dividing the forward to backward reaction, we get 10 to power 2, which is equal to 100. So the equilibrium constant for this enzyme catalyzed reaction is 100. The correct answer is option 1. Okay, let's move to the next question. Question number 18. What kind of inheritance is indicative in the pedigree chart shown below? So let's see whether it is a dominant or it is recessive. So it is not skipped any of the generation, so it is dominant. So option 3 it is wrong. Is it the Y-linked? No, because the female is also affected. So option 1 is also wrong. So it may be, the correct answer is maybe the option 2 or option 4. It is X-linked dominant or autosomal dominant. Let's see, it is a X-linked dominant. Okay, if it is X-linked dominant, then the father is infected. Here the father is infected. If father is infected, then the children should be infected, but neither of them are affected. So this one is also wrong. So the correct answer is option 4, that is autosomal dominant. Okay. So we can simplify like this to solve the pedigree chart. Which of the following RNAs possesses the peptidyl transferase activity? Peptidyl transferase activity, it is present in the larger subunit of ribosome. It is present in the larger subunit of the ribosome, and this activity is done by the rRNA. So let's see which one is the larger subunit. Larger subunit of RNA is 23S rRNA. This question is most oftenly asked in CSIR net question, and this is very simple question from molecular biology, and the correct answer is option 4, that is 23S rRNA has peptidyl transferase activity. Okay. Let's see the next question. Which of the following statements about human lines, that is long interspersed nuclear element, is false? Okay. Let's see the options. Lines or the long interspersed nuclear element is located primarily in euchromatic regions. Yes, both the lines and the signs, that is the sign is the short interspersed nuclear element, they are located in the euchromatic regions. This is the correct answer. Lines cannot encode all the product needed for their retrotransposition and depends on sign for some components. This one is wrong, because the lines are the autonomous, but the signs, they are non-autonomous, and for the retrotransposition activity they depend on line. Okay, so here the sign is depending on the line, not the line depending on the sign. So this one is wrong. Option 3. One of the three human line families, line 1 is the only family to have actively transposing members. Yes, this one is correct. Active line 1 element possesses an internal promoter located within the 5' untranslated region. This one is also correct. Okay. The wrong one is option 2. The false is option 2. Which of the following is a plant secondary metabolites? Okay. The secondary metabolites are the products which are produced by the plants, bacteria and the fungi, which are not directly involved in the development, growth and the reproduction. So which one of the following is the secondary metabolites? That is abbatic acid. Okay. Other three are the primary metabolites. So the correct answer is option 2. Next question is 23. Which one of the following signaling molecules is not a protein or a peptide? Transforming growth factor, TGF alpha. This one is the protein factor. Erythropoietin, it is a protein that is secreted from the kidney. Granulocytes colony stimulating factor. This one is also the protein, but epinephrine, this signaling molecule, it is synthesized by tyrosine, and this is not a protein or peptide. So this one is correct option, that is option 4. Question number 24. If an early cleavage stage wild type Drosophila embryo is injected with bicoid mRNA at the posterior pole, then. So this question is from Find It, Lose It, Move It experimentation. I do have a video on this topic, so please do watch that video. So if an early cleavage stage wild type Drosophila embryo, it is injected with bicoid mRNA at the posterior end. So bicoid mRNA, it is present always in the anterior end, but it is injected toward the posterior end. Okay, then what will happen? The head structure develop at posterior pole, while the same is inhibited at the anterior pole. Why it will be inhibited at the anterior pole? Because in the anterior region always the bicoid is present, and that forms the head. So it will form the head at the anterior end. Also the head, it will be formed at the posterior end as the bicoid is injected toward the posterior end. Head structure formed at both the poles. Yes, this one is correct. Head structure are not formed at the posterior pole due to the presence of posterior morphogens. If the posterior morphogens are present, but because of the presence of the bicoid, it will form the head on the posterior end also. So this one is wrong. Duplication of usual posterior structure occurs. This one is wrong. So the correct answer is head structure. It will be formed at both the poles. It is option 2. Question number 25. Which one of the following essential plant mineral nutrients complex with mannitol, mannan, polymanuric acid and other constituents of cell wall, that is boron. So this one is the direct question. So if you know it, then you can answer it. Otherwise, it's not wise to answer this type of question. And the correct answer for this question is 4. Question number 26. Which one of the following represents the largest outflux of nitrogen from the atmospheric reservoir? It is the very simple question and the answer is biological nitrogen fixation. I think this question does not require any explanation. So the correct answer is option 1. Question number 27. If the blastomeres of 4-celled sea urchin embryo are isolated, each blastomere can form a pluteus larva. This is an example of, if the blastomeres of 4-celled sea urchin are isolated, then each one it will form the pluteus larva. So this is a autonomous specification. Okay, let's see all the options: autonomous specification, conditional specification, determination, and mosaic development. Okay, so if it is autonomous specification, then autonomous specification are the determined, and autonomous specification always lead to mosaic development. So this is the type of question you have to find the odd one out. So which one is odd? That is the conditional specification. This is the example of an conditional specification. So the correct answer is option 2. Okay. So in part B, mostly the question are from the basics. If your basic is clear, then you can answer the question by simple analysis. So let's move to the next question. Question number 28. The following statements describe the outcome of genetic drift. Okay. Genetic drift, it is the change in allelic frequency in a population from generation to generation. So, which of the following statement describes the genetic drift, outcome of genetic drift? So, genetic drift, it may result in loss of some alleles, including the beneficial ones, and it takes the some other alleles. So, this one is correct. Genetic drift can be associated with population bottleneck. Population bottleneck and founder effects are the examples of genetic drift. So, genetic drift, it is associated with population bottleneck. Option D, genetic drift, it is associated with founder effect. So, option B and option D, these are also correct. Let's see the option C. Genetic drift is not observed in population that increase in size once they grow through a bottleneck. This one is wrong because the genetic drift, it is seen in the bottleneck population. So this one is wrong. So it is asking about which one of the following combinations represent all correct statements. The correct statements are option A, B and D. It is option 3. Option 3 is the correct answer. Question number 29. Which one of the following is a correct statement about the difference between a F plus and HFR cells? So in bacteria, F plus cell, and let's see, in the bacteria, this one is HFR. HFR is the high frequency recombinant. So this is the bacterial chromosome. Here the chromosome is also present. It is called the F plus cell because of the presence of F plasmid. Okay, this one is F plasmid. It is called the F plasmid because the plasmid, it contains the F factor, that is the fertility factor. And this F factor, it is present sometime independent or sometime it may be integrated into the main chromosome. So it is also known as the episome. Okay. In the F plus cell, it is present independently of the chromosome, and high frequency recombinant, it is a derivative of F plus cell where the F factor, it is integrated into the main chromosome. It is integrated into the main chromosome. So let's see which of the following is correct statement about the difference between F plus cell and HA cell. That is option 4. Option 4, here it is written in F plus cell, F factor is in the form of an episome. Okay, it is present in the form of an episome, while in HA cell, F factor is integrated to the bacterial chromosome. Here it is integrated into bacterial chromosome. Let's see the other option. In F plus cell, F factor is an integral part of bacterial chromosome. No, it is not an integral part of bacterial chromosome. While in HFR cell, F factor is present in episome. It's just the reverse. So this one is wrong. F plus and HFR cells are synonymous. Never. This one is also wrong. In F plus cell, bacteriophage carries F factor. Bacteriophage carries F factor. So this one is also wrong. So the correct answer is option 4. In F plus cells, F factor is present in the form of an episome, while in HFR cell, F factor it is integrated into bacterial chromosome. Okay, this is very simple question from genetics. Okay, question number 30. The common cuckoo, a parasitic bird, lays egg in the nest of other bird species. Soon after the cuckoo egg hatches, the chick shoves the nest owner's egg out of the nest. This is an example of. Let's see the option. First one is habituation. What is habituation? Habituation means the condition of being habituated. Okay. So this one is not correct. Then what is imprinting? Imprinting is a type of learning behavior that takes place during an early life of animal, or it is called the phase sensitive learning period, or it takes place during the critical period, right? So this one is also not. Innate behavior or instinctive behavior. Yes, this is the innate behavior or the instinctive behavior. Okay. And operant conditioning. So operant conditioning is a learning process or learning behavior where the behavior it is modified, or the behavior that depends on the reward and the punishment. So this one is also wrong. So the correct answer is option 3. Thank you so much for watching this video. So it's very important to know the explanation of each of the questions. It will help you definitely in future. In the coming session, I will explain questions from 31 to 50. So guys, please be stay tuned. Bye bye. See you in my next video. And if you are new to my channel and have not subscribed my channel yet, then please do subscribe and don't forget to share with your friends.